All right, going to share my screen here. All right, everyone see this main slide? Nico, nod if you can see it.
I can see it. I can nod, but my camera is deactivated.
Okay, amazing. All righty. Well, without further ado, I will begin. Hello, everyone, and thank you for being here to witness my thesis presentation entitled In Situ Weathering of Calcium-Bearing Minerals in Forested Ecosystems. Some of this intro is going to be a little bit like deja vu for my committee and others who were here for my proposal and progress report presentations, but hold tight because there is a slew of new information. Now, more than 40% of the total land in the Northeast is forest. It is, in fact, the most forested part of our country, but it likely won't be this way forever and perhaps sooner than we think. Northeastern forests serve as areas for locals to recreate. It stimulates the local economies by offering scenic views to vacationers and provides timber harvests, which employ longtime residents. But this rural beauty is at stake, and with it, the environmental and economic stability of the Northeast. Going to take you through a run through of the structure of this year thesis of mine. Introduction, what we expect to begin with. I'll introduce you to calcium depletion, some facts about soil min, and introduce my research questions. We'll get into my methods, kind of split up by soil mineralogy and in situ weathering. You'll know what those mean by the end of this presentation. We'll get into results and discussion for those same objectives. And then I will discuss my conclusions and the overall implications of this research. But first, some quick background on nutrient sources. First, when I think of trees, I often get a flashback to second grade science, where I learned about photosynthesis for the first time. And photosynthesis is the most extensive biosynthesis process on Earth, and trees perform this process to gain energy to grow, develop, and reproduce. But trees don't get the nutrients they need through this process, as chemical energy produced is just stored in the form of glucose and other carbohydrates. But trees in the Northeast primarily source their nutrients from the soil that they're grown in. And the soil contains both inorganic and organic types of vital nutrients like nitrogen, phosphorus, potassium, and calcium. In fact, without nitrogen and phosphorus, photosynthesis would not carry out sustainably. And nutrients like calcium, for example, are very important as they indirectly influence photosynthesis by maintaining cell wall structure and act to signal the opening and closing of stomata. And this research is really to highlight this inorganic source of nutrients for trees, the parent material or the rocks which the soil was developed from, which in turn shapes the mineralogy of the soil. And I have these two clauses down here, fungal associations and root exudates, since both directly and indirectly aid trees in accessing these inorganic and organic nutrient sources. And so we'll dive deeper into those later on in the presentation. I do want to note that I took this photo and it's my favorite photo from fieldwork 2 years ago now. And an example of the importance of these nutrients for trees is highlighted in a 2015 study by Hallman and others. And this is research that is deeply emotional for me because I'm obsessed with local Vermont maple syrup. But sadly, the study found that reductions in calcium availability and increases in aluminum availability in New Hampshire soils related to climate change and pollution, which I'll get into on the next slide, was associated with high rates of mortality for the sugar maple species, which will ultimately mean less maple production in the region. So our little maple man is unfortunately deceased. And to further this idea, historical nutrient depletion is putting the health of northeastern forests at risk. The acid rain era is defined by industrialization in the 1900s. And during this time, large amounts of sulfur and nitrous oxides were emitted from facilities in the Midwest, brought to the Northeast by the westerly winds, and precipitated out as acid rain, causing intense acidification of soils and associated nutrient depletion. Now, this acid deposition peaked around 1970 with the passing of the Clean Air Act, but some studies show we are still seeing negative effects from this period, and it's important to gauge if and how soils may be recovering. Climate change can also contribute to soil nutrient depletion. So under a changing climate, temperature, precipitation, and biogeochemical cycles are interacting in these new ways that are highly likely to cause a net decrease in key nutrients in northeastern soils, which will ultimately limit the growth of forests and overall forest productivity. For example, with increasing precipitation, there's more chances for water to percolate through a soil profile, dissolve key nutrients, and exfiltrate from a forest nutrient system. And we can't be making this any worse, right? Unfortunately, humans tend to make environmental issues incredibly worse. So increasing demand for wood products and biofuels is influencing more intense harvest regimes, which are further depleting soils. And we often think about the transition from fossil fuels to biofuels in a positive context, but what was once depletion from acidic industrial and fossil fuel emissions may transition to depletion from excessive tree harvest to produce biofuels. More and more of this nutrient-rich wood is being taken out of a typical forest nutrient cycling system, which decreases the amount of nutrients able to decompose and recycle back into a soil profile. And with less residues like branches and leaves on the forest floor, the soil profile is more susceptible to erosional processes like water percolation and biotic disturbances, which in turn leads to more nutrient depletion. And because of this disturbance in the nutrient cycle, older forests are showing less and less mass than previously gauged, and forests are not able to withstand this nutrient shift in a way that's sustainable for further intense harvests. And this is a depletion problem that can be compounded by forest ecosystem aging. If you think about it, this makes sense. Many studies suggesting that for some tree species, there's a general increase in the amount of calcium required for older stands to sustain that large, fluffy biomass, especially in response to human-caused stressors like the ones that I presented. And in forested ecosystems, the depletion of base cation nutrients, calcium, magnesium, and potassium can impact forest longevity. Though the growth of vegetation has been historically linked to nitrogen and phosphorus availability, recent declines in the growth and regeneration of key species like the sugar maple that I showed you before in the northeastern US have been linked to base cation depletion associated with acid deposition. So the weathering and subsequent availability of minerals containing these base cations is critical as they contribute to neutralize acids imported into the forest ecosystems by atmospheric deposition. And since calcium 2+ is the most abundant base cation in these soils, the weathering behavior of calcium-bearing minerals is of particular significance. And calcium really serves as a key species limiting nutrient, according to many studies, having been correlated with drops in phosphorus. But studies indicate it will be the first base cation to fully deplete from soil available pools. And so in combination with acid deposition, nutrient export accompanying timber harvest could lead to a reduction in total soil and biomass calcium by 20 to 60% in only 120 years. And so here's our little deceased maple man to remind you of the New Hampshire sugar maple study. But let's get into the mineralogy of it all. Apatite, anorthite, and calcite in soils are critical providers of inorganic calcium through weathering, but continued stress on the environment could cause cation depletion rates to surpass rates of weathering, leading to a net export of calcium. And as the main source of inorganic phosphorus in nature, apatite dissolution is of growing interest to gauge longevity of forests in the Northeast. It's relatively insoluble at a neutral pH, and the degree of dissolution and solubility intensifies with acidity as expected. And this equation 1 is kind of a general dissolution reaction for apatite in the presence of acids. And then, as a calcium-rich feldspar, anorthite is one of the biggest contributors of calcium to soils, making it integral to soil fertility and forest productivity. And in acidic conditions, calcium and aluminum can be desorbed from the mineral surface, leading to the formation of edge pits. The protonation of anorthite can also result in the formation of clay minerals like kaolinite and gibbsite, for example. And here is another just general dissolution reaction for anorthite in the presence of acids. Now, calcite exhibits a notably high rate of weathering in typical soils, deeming it the smallest in relative abundance of soil mineralogy in northeastern forests. And the high solubility of calcite makes it more susceptible to alteration with increasing acidity relative to these other two calcium-bearing minerals. And of note, moving forward, it's really important to note that the rates of weathering for all these minerals are influenced heavily by three main factors. The first being reactive surface area, so the amount of area available for weathering of each mineral. Organic acids produced by microorganisms in the environment. We'll get more into that later. And presumably the overall concentration and distribution of chemical elements in the parent material, which you will know a lot about by the close of this presentation. But going forward, for calcium-bearing minerals to supply ecosystems with available and exchangeable calcium, they've got to be weathered. And in soils, mineral grains are subjected to a variety of physical and chemical processes driven by both abiotic and biotic facilitators. And when examining soil mineral calcium budget, it's important to assess these prominent biogeochemical mechanisms driving availability. And this can be accomplished by the analysis of associated surface textures at the micro to nano scale. So in this figure, I'm going to walk you through some examples of these diverse mechanisms. But in A, we have a photomicrograph of a pristine anorthite surface, but let's see what can happen when a mineral like this is subjected to biotic weathering. In B, we can see a fungal hypha actually penetrating a surface layer sheet of biotite. This is an example of biomechanical weathering. That hypha is just kind of getting in between the layers of that biotite right there. But what about some abiotic weathering? Here we see a rough apatite grain surface covered in etch pits, and this could suggest intense proton attack and depletion by acid deposition. And another example in C, here's an apatite surface covered with molds and spherical aggregates, another example of biotic weathering mechanisms. Now, these diverse transformations can both promote and inhibit calcium availability from primary minerals. And these transformations are, again, governed by those three main factors introduced on the last slide: the reactive surface area of the mineral, organic acids produced in the environment, and presumably parent material composition. Now, upon weathering, calcium from the mineral structure becomes calcium 2+ in soil solution. And from here, calcium can be taken up by plants. It can become immobilized by bacterial communities, absorbed onto cation exchange sites, precipitate out into neophases, like seen here in C. We've got some beautiful precipitates, or even exfiltrate with soil solution. So here are those diverse mechanisms, both making calcium available and immobilizing them, making them unavailable. And at the microscale, transformation of grain surface textures reflects these intricate processes and can be used to infer mechanisms for primary mineral weathering. And unfortunately, these analyses are often performed in lab settings, and discrepancies between lab experiments and in situ mineral weathering experiments have been widely observed. For example, I believe 3 of these were lab experiment related as opposed to in situ experiment, but we'll be doing in situ experimentation, which I will get further into. But first, I want to present my overarching questions. The first being, I sought out to address what are the cumulative effects of parent material composition and ecosystem aging on current nutrient dynamics in northeastern forest soils. And there's another tree growing in our forest here. The second objective of my thesis was to determine what may be the primary mechanisms driving mineral dissolution and inorganic calcium availability in northeastern forests. Now, I had some hypotheses here to begin with. The first being that soil parent material or glacial till source in the case of the Northeast drives acid buffering capacity across these sites and therefore calcium nutrient dynamics. I also said that ecosystem aging would have a lesser impact because if parent material calcium mineral content is what controls acid buffering capacity, the aging of ecosystems with nutrients supplied by various parent materials should be a secondary factor. And I also predicted that proton attack would be the primary mechanism of weathering in northeastern forests. And this would make sense considering historical and some current acid inputs, as well as the relatively little time it takes for acid to weather minerals as opposed to processes which take longer periods of time, like hydrolysis under normal pH, physical erosion, or even symbiotic weathering. But let's see what my results say later on. The following research was really conducted to address the knowledge gap of microscale weathering mechanisms required to gauge current soil nutrient dynamics in northeastern forests and to really assess soil recovery from historical and continued stressors discussed. And for this study, forests were chosen due to their long-term history of management and record keeping, their differences in underlying geology, and similar tree compositions. So the first site here, represented by COR, is Clement Woodlot Forest in Corinth, Vermont. It's a Dartmouth College property with some nearby contracts for private timber harvests. And the second site, represented by SCG, is Second College Grant in northern New Hampshire. It's also a Dartmouth College property with 27,000 acres or 46 square miles of forest. It is my favorite forest site. It's where I took that picture of the frog earlier. And the last is the heavily studied Bartlett Experimental Forest, represented by BEF, and it's located towards the center of New Hampshire. Now, to discern differences in nutrient dynamics with stand age class was determined via observed above ground biomass and basal area for common tree species. And so 15 stands at each site were chosen and composed of 5 stands per age class, being young, intermediate, and mature. So you can see all those plotted out here. And these sites represent a range of parent materials and related soil nutrient states. Using estimates from ice flow direction during the Pleistocene epoch and knowledge of the bedrock, we estimated the main source of till as representative of a wedge of material upstream of the site. Clement woodlot forest parent material is largely chalcopyrite, making it our presumed calcium-rich ecosystem. While Bartlett experimental forest parent material is mostly granite and metamorphic in composition, deeming it the calcium poor site. And second college grants parent material is the calcium nutrient intermediate between the two in terms of parent material. So from the above table, these sites again are largely similar in native tree composition, soil order, and elevation, but main differences are reflected in the parent material and exchangeable calcium. So we can see some exchangeable calcium values highlighted here. And exchangeable calcium was determined by collaborators at UMass Amherst using 2 grams of soil across multiple depths. And samples were oven dried and then extracted with ammonium chloride and shaken for 24 hours. The soil slurries were then centrifuged and filtered to 0.45 micrometers. And samples were then analyzed for exchangeable calcium concentrations using the ICP-OES, or the inductively coupled plasma optical emission spectrometer at UMass Amherst. I dare you to say that 5 times fast. ICP results were calibrated using a 9-point standard curve. An average exchangeable calcium at each forest site was determined by averaging results from 170 soil samples across steps, ranging from the forest floor to 100 centimeters below the surface. And average exchangeable calcium really highlighted the variable nutrient states ideal for comparing these weathering features for native calcium-bearing minerals across forests. So you can see how high it is here at COR at 967 milligrams per kilogram of soil compared to 1/3 of that value at BEF and an intermediate value at SCG here. Now, to test my hypotheses, I've created a set of objectives in order to keep my analyses organized. And the first objective was to identify and quantify calcium nutrient pools in soils at our study sites. And this will help verify the degree to which parent material influences soil calcium nutrient state. And this was done by comparing values for deep soil mineral calcium, exchangeable calcium, and total calcium. While the second objective involved characterizing mechanisms for in situ weathering. And this was to test the hypothesis that parent material calcium is the primary driver of acid buffering capacity across sites, and that proton attack is the primary weathering mechanism for inorganic calcium availability. Now, to accomplish this, calcium-bearing minerals were left to weather in situ for a year, and after collection, I performed SEM imaging and semi-quantitative review of weathering features distributed on mineral surfaces. Lysimeters were also installed, for which I have soil solution, calcium, phosphorus, aluminum, and iron values to aid interpretations of what we're seeing at the mineral surface. I'm now going to kind of go through more in-depth techniques, results, discussion, and conclusions for the first objective here on the left, before transitioning to the same for objective 2, and then I'll bring them both together to discuss the overall implications of this research. Now, to accomplish the first objective, I have employed multiple techniques. At each site, 9 soil pits were excavated to a depth of 80 to 90 centimeters, and soil samples were acquired at the same depth, which corresponded closely to the unaltered parent material's calcium nutrient pool. And soils were excavated from the side of the soil pits and stored in plastic bags in the dark until analysis. And so at each study area, I have quantified calcium-bearing minerals via X-ray diffraction analysis using the Rietveld refinement module. And each of the 9 soil samples took about an hour or so to run, and another, on average, 2 hours to kind of refine in the program. And so 3 representative samples were then chosen, 1 for each site, to compare with averages for 3 deep soil exchangeable calcium and total calcium values per site. So you'll see that in our results. And exchangeable calcium and total calcium were both determined by the PhD on our research grant, Alex Rice, using the ICP-OES I mentioned before. And the method for processing soils for exchangeable calcium involved drying soils, extracting them with ammonium chloride, shaking them for 24 hours, then centrifuging the samples and filtering them. And the method for processing total calcium involved digesting the samples with nitric and hydrofluoric acid, heating to 170 degrees for 48 hours, drying the blend to a paste, then resuspending the paste with nitric acid and drying them down once again. And this procedure was repeated a final time before, again, analysis with the ICP. But let's get into some mineral calcium results. So soil mineralogy indicated the pool of calcium-bearing minerals didn't appear to vary too significantly, but relative abundance of these minerals did. So while generally dominated by quartz, the parent material really differed in content of feldspars, mica, and clay minerals. And most importantly, each parent material had a different signature with respect to phyllosilicates and feldspars, with Corinth here largely dominated by quartz. We could see that big 60.4% mineral abundance. And BEF with great amounts of feldspars, unexpectedly containing the highest amount of calcium plagioclase and calcium-bearing phases detected of the 3 sites. And to remind you, BEF is the calcium pore with respect to parent material, surficial geology, aka the glacial till source that we saw, but I'll explain why that might be. And second college grant here containing large quantities of micas. Now, on the right of this graph, you can also see corresponding soil composition oxide estimates calculated by throwing the mineral abundance percents on the left into the USGS HandLens computer program. Now, no apatite or calcite was detected in the parent material, which isn't to say it's not there. X-ray diffraction is limited in detecting minerals that are low in abundance and size relative to other minerals more commonly found in the soil. So this is a glaring limitation of X-ray diffraction, which could account for the surprisingly high calcium values you can see here at BEF. But before we get more into that, let's see how exchangeable and total calcium differs. Average exchangeable calcium from deep soil samples highlights the amount of mobile calcium, while total calcium values takes into account all calcium in the soil, aka both exchangeable and immobile mineral calcium. And please note the different units between exchangeable and total calcium values, one being a milligram per kilogram of soil and one being a milligram per gram of soil. Now, at COR, deep soil exchangeable calcium is orders of magnitude above that of BEF and SEG, only about an order of magnitude from SEG and 2 orders of magnitude from BEF. And this difference isn't so apparent when comparing total calcium across sites. Total calcium was highest at COR as well, but relatively similar at BEF and SCG. And it's also important to note that despite the 80 to 90 centimeter depth of these values, they still can be influenced by soil texture and weathering and erosion rates of the topsoil. For example, soils with high clay content typically have higher amounts of exchangeable calcium due to higher absorption capacities from those negative charges that sit on clay minerals. Now to discussion. Soil X-ray diffractograms indicated that the pool of calcium-bearing minerals didn't appear, again, to vary too significantly, but we saw that the abundances did. And it's important to note that this method does not account for trace minerals. In particular, the expected chalcopyrite nature of the parent material at COR was not reflected in the mineralogy of the soil that from XRD analysis. But as Bailey and others indicated in 2019, calcite and apatite are too small and low in abundance to be quantified with X-ray diffraction alone. And this justifies the use of more advanced EPMA characterization, like the cluster analysis developed by the same group to identify major calcium contributors in soils. And we are currently in the process of doing this characterization, and data is to come out in a future published paper, so keep an eye out for that. But luckily, differences in calcium nutrients are captured in total and exchangeable calcium data, where both deep soil total calcium and available calcium nutrients were greatest at COR. Discrepancy in calcium budget between XRD and total digest and exchangeable calcium indicates that probably most calcium is present in the non-mineral pool, aka mobilized or organic, or that X-ray quantification is just not accurate enough to detect those trace calcium-bearing minerals that I've been talking about, calcite and apatite. But overall, our results indicate that the state of calcium in studied soils does reflect the sourcing of the till material, with the dominant effect of chalcopyrite on the calcium budget in COR. And this emphasizes the clear parent material control on nutrient availability in the studied forest soils. But now that we're done with objective 1, I'm going to transition to our mineral weathering objective. For the second objective concerning the mechanisms for in situ weathering, techniques were focused on examining the weathering behaviors of specific calcium-bearing minerals. And so I introduced 3 grams of previously characterized ground anorthite, apatite, and calcite powders to soil profiles at each forest. And these 200 micrometer mineral powders contained by 5 micron mesh bags were left to weather in the field for a year at a depth of 30 centimeters. And the mesh bags allow for natural weathering processes to act on the minerals without roots breaking the bags apart. And since I seek to address how current nutrient dynamics are influenced by ecosystem aging, bags were installed at young, intermediate, and mature stands at each forest site. So this video that keeps playing in the background is a video of my mesh bag installation a whole 2 years ago, which feels like yesterday. Of note, lysimeters were also installed at each of the forest sites about 50 centimeters below the surface, and water was collected about twice a year during the late spring and fall seasons using a vacuum pump by the amazing Alex Rice once again. Alex then got pH values for samples before analyzing the soil solution water by first acidifying samples with nitric acid, before running them on the same ICP OES mentioned in Objective 1 to get values of soil solution calcium, phosphorus, aluminum, iron in milligrams per kilogram of water. And this is really to help with the interpretation of what we're seeing at the mineral surface and overall characterization of mechanisms for this objective. Now, scanning electron microscopy provided high-resolution micrographs of grain surfaces needed to gauge in situ weathering intensities of apatite, anorthite, and calcite. You can see these pre-weathered minerals here that were used and compared to post-weathering surfaces that you'll see in the results. And after 1 year of in situ weathering, mineral bags deployed at each forest site were collected, 81 samples in total, and upon collection, mineral bags were assessed for damage and weighed. The nylon mesh bags were cut open and powders were weighed separate from mineral bags prior to being stored in individual vials away from heat and light. From here, 27 representative samples, I got that by multiplying 3 forests, 3 sand ages, and 3 minerals, were mounted on carbon tape, sputter coated then with gold and palladium. And then I spent many hours in the microscopy lab taking photomicrographs with the SEM and performing a precipitate analysis with the EDS detector attachment. And so in total, I looked at about 243 SEM photomicrographs as I looked at 3 grains within each of the 27 representative samples and took anywhere from 2 to 5 photomicrographs per grain. And first, I want to talk about the soil solution chemistry results. So this is the results from lysimeters. And soil water composition for the 3 study sites revealed significant differences in terms of their pH and concentrations of calcium, phosphorus, aluminum, and iron. For example, soil solution at COR had the highest pH of the 3 sites and significantly higher concentrations of calcium. And this confirms that the soils at COR are less acidic than those at BEF and SCG and have higher calcium nutrient availabilities. Now, soil solution at BEF had the lowest pH of the 3 sites and lowest concentrations of calcium and phosphorus, showing that the site is relatively acidic and has lesser availability of these historically limiting nutrients. And interestingly, at SCG, soil solution showed similar pH and phosphorus levels to that of BEF, but notably higher concentrations of calcium, aluminum, and iron, again really highlighting the site as the intermediate of the 3 in terms of nutrient abundance. And these elemental compositions of soil solution also differed kind of considerably across stand age. For example, at COR, soil solution concentration of calcium was highest in the young and intermediate stand. However, the mature stand had a significantly lower concentration of calcium witnessed. And so the concentrations of aluminum and iron in soil solution were low and relatively stable, if not a bit of an increase at the mature stands, while phosphorus appeared to increase with stand age in this beautiful step-like pattern. And soil solution concentration did not vary as a function of stand age at BEF. At this site, concentrations of calcium were consistently low across all stand ages, and concentrations of aluminum and iron were variable with apparent jumps at the intermediate stands, while phosphorus generally remained stable. And at SCG, soil solution concentrations in aluminum, phosphorus, and iron consistently increased with stand age in that beautiful step-like pattern, while calcium remained within a similar range, if not decreasing slightly. And it's important to note the degree of variability that comes with in situ experimentation, highlighted in some of these more elevated standard errors. And now that we've got soil solution results from lysimeters out of the way, I'm going to introduce the weathering features I observed on these minerals after a year of weathering at each forest site, starting with etch pit features. The style of etch pitting features differed significantly by forest site and by mineral species. And pit shapes largely depend on mineral grain orientation, while the number of pits has been correlated with dislocation density, according to some materials studies. But with that in mind, at COR, there were a few incipient etch pits observed on anorthite grains, and you can see that here and a little bit here, with no notable observations on apatite or calcite grains. And these shallow strip-like pits appear to represent initial stage pitting processes. And the shape should kind of remind you of this image from Objective 2 up here. It's almost perfectly like that image that I made. While in direct contrast, calcite mineral grains deployed at BEF exhibited this intense cleavage-defined etch pitting, dislocations in which uniformity seemed to correlate with stand age, which we'll get into more later. But you can see that the young, intermediate, and mature seems to kind of go up in these intensities. And notable strip-like pitting, similar to what we saw at COR on the previous slide, was also observed on anorthite and apatite grains, but they seemed relatively higher in frequency and intensity. And so it's kind of details a higher degree of likely abiotic mediated dislocation at this site compared to COR. While at SCG, edge pitting wasn't really seen on apatite or anorthite grains. However, calcite grains displayed terrace pits that were spatially isolated across surfaces, which seem more biotic in origin. So you could see this hyphae attached to the surface, and the pits kind of correspond to where the hyphae locations are, likely from the excretion of acids to get at that mineral nutrients. And so you'll see me discuss biotic features in a minute, but first we are going to dive into some precipitate analysis that I performed with the EDS detector. So EDS analysis revealed elemental compositions of select surface precipitates, which seem to vary also by study area and mineral species. At COR, anorthite grains had these clay-like precipitates characterized by this plate-like geometry and signature potassium, aluminum, and iron peaks here in the EDS spectra. And some apatite grains also showed evidence of dissolution occurring in spaces between mineral particles. So you could kind of see that texture here. I did block it a little bit with my X, but it produced this unique texture that didn't significantly alter the bulk mineral surface. And you could see that here in this. It looks almost identical to the apatite formula. And this is a phenomenon known as interstitial dissolution. And in apatite grains at this site, it seemed to result in the reprecipitation of apatite from solution. So kind of cool to see that here. Was not expecting that texture. And at BEF, anorthite displayed the most notable precipitation processes of the three mineral species, where EDS spectra revealed compositions of both iron oxide coatings here and ferromagnesian clays. So you can kind of see those oxide coatings on that aggregate here, as well as some clays, some matted clay formation over here, and the associated spectra. And at SCG, calcite grain surfaces were frequently covered by ferromagnesian clay precipitates, appearing a bit thicker and more layered than those observed on anorthite surfaces at BEF. And you can kind of see that here. Again, seems to be a little aggregate with these clay formation. And there was clay formation up here with some matted layering up as you go up on this mineral surface. And oh, hello, there's hyphae again. They can't escape. We can't escape the hyphae. But we're going to talk about the hyphae now. Biotic weathering features observed, like hyphae and biofilm, it appeared that the presence and type kind of appeared to differ across sites and mineral type. And for example, at COR, we usually begin these with COR. COR is the start, but we're starting with BF now because we did not see any evidence for biotically mediated weathering processes at COR. So that's sad, but we do see some at BEF to start. And it's actually notable and intriguing. The colonization of calcite and apatite grains differed pretty significantly at BEF and SCG. For example, here, calcite grains at BEF showed no evidence for hyphae or associated biofilm. These are all apatite. We did not witness a single hyphae interested in calcite at BEF, but you can see that apatite surfaces here had some hyphae at the surface trying to colonize, maybe forming a little bit of biofilm and on top of this aggregate here, just trying to get those nutrients. The phosphorus probably. But the opposite is true for SCG. Calcite surfaces here showing this intense fungal colonization at the mineral surface, even with presumed matted layers of this biofilm, with few hyphae present on apatite surfaces deployed at this same site. So that's a major difference, which we will talk about ongoing. But the frequency analysis of these features after weathering revealed distinct relationships across forest sites and mineral species. And I don't expect you to remember all of those SEM photomicrographs from the previous slides, so this graph kind of captures them in a pretty summary form. And edge pitting, to start, was predominantly observed on mineral surfaces deployed at SCG, composing 27% of all observed features. While BEF also had a high rate of edge pitting at the surface, accounting for 21% of all features, witnessed at the site. And at COR, dislocation features only accounted for about 10% of observed morphologies, with only 4 total observations. And I had showed you 2 of them or so, and 1 on an apatite surface deployed here. And with respect to mineral species, calcite surfaces were the most likely to display edge pitting across BEF and SCG, accounting for 50% and 37% of calcite observations at each site, respectively. And precipitation processes were the predominant form of alteration across all forest sites, representing 81% of features at COR, 47% at BEF, and 42% at SCG. And at COR, 100% of apatite surface observations were precipitates. And I want to remind you of that interstitial dissolution texture that we saw before. It all looked like that. It was all those very cool interstitial apatite reprecipitating from solution textures that we observed, while precipitates were most likely to be observed on anorthite at BEF and SCG, accounting for 51% and 74% of all feature types observed on the mineral, respectively. Now, the occurrence of hyphae in biofilm was the least observed alteration across all forest sites, composing 8%, 32%, and 39% of all features observed on COR, BEF, and SCG, respectively. And out of all mineral types for all forests, hyphae was most notably observed colonizing apatite surfaces, comprising 41% of morphological observations against 20% for anorthite and 26% for calcite. And distinct differences in biotic weathering processes are noted between calcite and apatite for BEF and SCG. Again, I had introduced that before. But at BEF, for example, 51% of apatite surfaces had organism-mediated surface features, with 0% on calcite grains at BEF. At BEF, if you can remember, the calcite grains were really dominated by that edge pitting, that rhombohedral edge pitting, but we saw no hyphae. But in contrast, 37% of apatite surfaces at SCG showed evidence for biotic influence, hyphae in biofilm, while 40% of calcite grain observations were likely biotic in origin. We saw those hyphae trying to colonize those surfaces of calcite. And these numbers contribute to the earlier idea that phosphorus content and base cation abundance in northeastern soils are both key to fungal colonization of apatite and associated weathering rates. And so on average, I want to note that the presence of these features also increased with increasing stand age at both BEF and SCG, but we'll dive more into that in the discussion. Now, for the initial discussion, I want to talk about how the range of paramaterials and associated soil nutrient states across sites appeared to greatly influence the intensity of weathering features driven by both abiotic and biotic facilitators. But first, I want to focus on abiotic because biotic can be a bit more complex. Now, at the start of experimentation, we knew that paramaterial calcium content increased in an ideal gradient across sites, with BEF at the low end and COR with the most paramaterial calcium. And during this research, we found that soil solution calcium, exchangeable calcium, and pH of the soil also followed this increasing trend, highlighting the strong influence of paramaterial on soil nutrient states at these sites. And this calcium data, along with the SEM analysis of weathering features, allowed for the interpretation of inorganic calcium weathering intensity and related acid buffering capacity. And to remind you, COR, again, presented the least extensively developed weathering features of the 3 sites, and it had the highest paramaterial calcium content, soil solution calcium, soil solution pH, and soil exchangeable calcium levels. While SCG revealed abiotic weathering features that were more medium in intensity relative to the other sites, and it had the mid-level of these calcium nutrient parameters. To remind you, edge pitting features on calcite here were deeper and more isolated than those at BEF. And the calcium supply could have impacted the equilibrium of calcite sufficiently to decrease the susceptibility of calcite at the site to weathering. While mineral surfaces at BEF showed the greatest overall abiotic weathering intensity and lowest calcium nutrient parameters. And so again, that uniform rhombohedral cleavage-defined edge pitting across calcite grains deployed here suggests that proton attack is the principal weathering mechanism. And the notion of proton attack being a dominant process at the site is further supported by the presence of iron oxide coatings on some anorthite grains that I had showed you in the EDS analysis. Now, the high frequency of biotic features observed on apatite grains at BEF compared to the lack of these features on calcite grains suggests that the presence of phosphorus in the mineral at the site was really favored the fungal colonization and related weathering intensities of apatite over calcite and anorthite. And previous research has indicated that mycorrhizal fungi can enhance the colonization and weathering of apatite under nutrient-limited conditions like phosphorus limitation or base cation limitation, as shown in studies by Blum and others in 2002 and another study by Leake and others in 2008. However, soil solution phosphorus concentration at BEF was really similar to that of SEG, with average values of 0.14 and 0.17 milligrams per kilogram, respectively. And fungal colonization was not dominant on apatite at SCG. This suggests that perhaps some P limitation or base cation, well, we know that base cation limitation is less prevalent at SCG than at BEF. And weathering features could correspond to differences in mycorrhizal communities. For example, there are two primary groups of these mycorrhizal fungi that they're separated into, called ectomycorrhizal fungi and arbuscular mycorrhizal fungi. And hundreds of species within these groups have different capabilities and needs in terms of accessing inorganic nutrients. For example, one species may be more equipped to access the carbon and calcite as opposed to the phosphorus and apatite. But this site-specific response of the biota to the presence of discrete apatite grains that we had deployed here does confirm the key role of apatite as an important nutrient source in soils, despite its scarce abundance and high stability. And the greater presence of hyphae and biofilm on calcite surfaces here at SCG, coupled with the nearby isolated edge pits, suggests more organic mediated dislocation processes at the site than COR, and maybe even BEF. And notably, the non-uniform terraced edge pitting witnessed on several calcite grains at SCG, again, appeared to coincide with hyphal colonization at the mineral surface. So the SEM micrographs here show that it is possible that organic acids like oxalic or citric acids produced by those hyphal activity in general are causing calcium ions to release in soil solution, similar to observations from prior studies. Now, the effective stand age on abiotic mineral alteration is particularly evident when looking at the calcite mineral surfaces weathered in situ at the stand chronosequence at BEF. Edge pits, again, appeared more frequent and developed across calcite grain surfaces weathered at mature stands here compared to the isolated shallow pitting covering calcite grains inserted at the young stand here. And for a direct comparison, I also want to show you a chronosequence of calcite at SCG. And the weathering intensity appears to increase slightly with increasing stand age, but with respect to the abiotic weathering features at the mineral surface, it's not as significant or apparent as with what we saw on the previous slide at BEF. And you can see here, again, this is another set of calcite, just for example, of photomicrographs with increasing stand age, but it's of note, it's kind of difficult to compare intensity. Considering calcite at the site, and you can see the hyphae, it appears to be more influenced by biotic alteration via organic acids. But there is evidence that proton attack from acids not generated by hyphae are also at play, and you can kind of see that here in the more uniform pitting at the young stand. And a weathering intensity gradient across aging soils was not apparent at all at the calcium abundant Corinth. And so this suggests that calcium deficient soils supporting more mature stands appear more reliant on inorganic calcium nutrient sources. And the weathering intensity with stand age appears truly compounded by the parent material calcium content, since weathering intensity with stand age was very, very clear at the calcium deficient site, less distinct at the intermediate, and not observed at all at the calcium abundant site. And this kind of supports a conclusion made by Hamburg and others, and they performed the study at HBEF, which is very close to our BEF site, who suggested that younger stands are better at and tend towards mobilizing calcium from typically harder to access organically bound nutrient pools, like decomposing leaf litter, than older ones who turn towards more soluble inorganic sources of calcium, like that of our minerals. And it is likely that nitrogen and phosphorus content that are key to this organic matter are really important and kind of drive this observation, as these are vital in the growth and development of the saplings that are at younger stands. And supporting a conclusion hypothesized by Yanai and others in 2005, the relative lack of weathering feature development on mineral surfaces at the calcium abundant COR one compared to the other sites indicates that the paramaterial calcium content is an important indicator when assessing calcium depletion risk with historical acid rain, intense harvest regimes, and climate change. And it appears that ecosystem aging also played a role in the type and intensity of weathering, affecting calcium-rich minerals. And in particular, the general increase in abiotic and biotic-mediated weathering features across chronosequences at least two of the sites, with the colonization of apatite at the calcium pore, somewhat phosphorus-limited BEF, and lesser colonization of this mineral at the nutrient intermediate SCG suggests that natural ecosystem aging also influences the colonization of apatite grains by microbial communities to meet plant needs. Also suggesting that older stands developed on calcium deficient soils may tend towards inorganic calcium sources as opposed to organically bound nutrients. And overall, this study suggests that parent-material calcium and phosphorus content are the primary controls on calcium mineral weathering in northeastern forests. And the influence of ecosystem aging shouldn't also be discarded as a secondary driver. Now, to verify this concluding hypothesis, additional data on nutrient concentrations of litter fall and additional root water sampling across stand age are going to be needed to confirm this influence of stand age on weathering intensity of these minerals in organic calcium. But some additional considerations and limitations include soil moisture and precipitation for one. Clement Woodlot Forest here was the calcium rich of the sites and it showed the least weathering. But it's important to note that soils at the site appeared drier than those of the other two sites, at least over the year of observations. When looking at the mesh bags after collection, the mineral powders were all very, very dry. Compared to those of the other two sites, which appeared to retain a good amount of moisture. You could see those hyphae kind of hanging onto the bag, and it definitely looks wet, and this is a dry bag. And this suggests that bags at COR remained mostly in unsaturated soil conditions, which could limit the effect and extent of kinetically controlled dissolution at this site specifically. And another consideration, I mentioned this earlier, but different microbial communities have different needs and capacities to access specific nutrients. So I did want to point that out again, because that's a very important consideration and could be the reason why we're seeing differences across sites. And it's very expensive to perform DNA sequencing on these hyphae attached. This is, I believe, hyphae attached to a root structure. So you can kind of see that hyphal network. And so yeah, we did with what we could in terms of, grant funding and whatnot. And we felt it was probably a little bit beyond the scope of this research to really do that DNA sequencing. And I also want to point out the variability in measurements that can come with in situ experimentation. Forest ecosystems are incredibly complex, and there are countless factors impacting what I presented today. And as many as possible in a 2-year period of research were considered. Now, this study is part of a larger USDA-funded project ran between UVM and UMass Amherst and involves several graduate and undergraduate theses focusing on nutrient cycling and managed forests. And so we've been working with public entities like the USDA Forest Service Northern Research Station, as well as private entities like Dartmouth College to conduct research on and around properties. And we're going to share our cumulative results and conclusions with forest managers from the Green Mountain and White Mountain National Forests, the states of Vermont and New Hampshire, and private timber industry to discuss our research outcomes and how it relates to their management activities, suggesting more sustainable methods with our results. And since our identification and quantification of mineral nutrients via XRD did not work as intended, as a reminder, it doesn't tend to pick up smaller, less abundant calcium minerals like apatite and calcite. This project is to make use of EPMA soil mineral maps, and there's a need for the cluster analysis developed by Bailey and others to accurately identify and quantify these mineral sources to complete a more accurate mineral nutrient budget for soils at each site. So keep an eye out for that paper, which will only help to enrich my current conclusions. And with any scientific venture I take on, I truly hope the results of this study can influence sustainable philosophies across the board. Thank you for hearing my thesis, the culmination of 2 years of life-changing, at least for me, work. And a special thank you to my advisor, Nico Pergriel, for affording me and guiding me through this opportunity to advance science. And a special thank you to Anthony D'Amato for providing access, guidance, and integral data on all study forests used in this research. And a huge, huge thank you to the summer fieldwork crew, spearheaded by UVM Field Tech Grace Smith, the UMass Amherst crew, including Project PI Justin Richardson, PhD candidate Alex Rice, Bailey, and Wen Chu, as well as Trevor McCowiak, Nico's prior master's student who's already moved on. And moving forward, I'm very excited to see our work get published. You could see Anthony here, Tony. He was definitely not photoshopped in this photo, but we're all having a great time. And some additional acknowledgements I have to go out to Siga and Sonia on the left here. They're the only other two people in my cohort, and they have become lifelong friends over the last 2 years. And so this is the first time we met at the bottom left, coupled with our most recent photo at the top from Texas. We just got back from spring break last week. It was so much fun. A bit warmer than Vermont, as you can imagine. My amazing goofy dad for reminding me not to take everything so seriously, as well as my twin sister and best friend for FaceTiming me every day to talk my problems out. My brother-in-law, Justin, for supporting my sister and I and always making us laugh, and for their service working in intelligence for the US Air Force. And of course, my dog Caesar for giving me kisses when I need them the most. Thank you so much for listening to my thesis.
Thank you so much. I see a lot of emoji, clapping emoji. You can also unmute yourself and clap. Thank you so much for this presentation. Thank you so much for attending this presentation. Fantastic slides. We'll move forward to the next part of the defense, which is to take questions from the audience. You can unmute yourself and ask the question, or you can write the questions in the chat. I will convey these questions as they appear. Congrats on a beautiful presentation, Vig. I love the pictures at the end. This is really putting everything together in terms of teamwork as well.
Yay.
We have a comment from Bella on the chat that says, excellent work, Vic. You've done a huge amount of work over these past couple of years. This is really impressive, with a heart from you. So yeah, the floor is open for questions. Feel free to ask them. I'll, of course, ask Justin, Julia, and Tony to refrain from asking questions at this point.
I have stunned them all.
It's always, I'm raising awkwardness at the questions moment. Leaving time for people to recover from this presentation.
Yeah, feel free to unmute yourself too if you just want to ask a question. There are no dumb questions. Ask everything.
There is a question that just appeared in the chat from Quinton Lewis. Of the mineral you placed in the ground, which was your favorite to observe and why?
Okay. Well, in terms of observations, and yeah, so I had a lot of favorite minerals before I started this research, just because mineralogy was my favorite subject in undergrad, still is my favorite subject in all of geology. Apatite was my favorite going into this project because I love green, and the color of apatite is more of like a green-blue. It has a cool prismatic habit when naturally formed. It doesn't have cleavage though, so that gives it negative points. But I have to say, after this research, calcite is my favorite of the three minerals because it had that beautiful rhombohedral cleavage. And after weathering, you could even see it more intensely after that, acid attack kind of getting at the mineral surface, and you could see those like rhombohedral kind of surfaces pop out even more. So it gets pretty cool. So yeah, that's my long nerdy answer to that question.
The right answer. I agree that the pictures of the calcite were pretty stunning. Any other question?
Hi, Isabella. I have a question. Vic, amazing job. I'm wondering, so I can't remember the name of the field site, which is why I was hesitant to ask, but you mentioned that one of them was really dry, potentially throughout the whole field sampling compared to the other ones. And so those bags, when you dug them up, did not retain very much moisture. Was that surprising to you?
I think it was, and when just looking back at the nutrient parameters for each site, or the parameters at each site, it did, in theory, get the lowest annual precipitation on average of the 3 sites, but not much lower than the other 2. They were all very similar in terms of annual precipitation. So it was surprising, I guess, to see that at the site. And the prior season hadn't been dry or anything like that. So again, it was surprising to see the minerals at 2 of the sites. The bags were all kind of wet, mucky, kind of water bogged a little bit, while those at COR were all dry upon being taken out of the soil. So it was a bit surprising, but in situ experimentation, there could be like some microenvironments forming, and other factors at play there as well. Thank you for your question.
Thanks.
Good question. Thank you. Any other question? Well, if there is no other questions from the audience, now is the moment where we will excuse the audience and ask questions as a committee to Victoria about experience, about her thesis, about her presentation. I want to thank again the audience for being there, for joining us. This was a great crew that joined us on this call. So thank you so much for coming. And we can also applaud Vic for her work again.
Thank you all for coming.
Yay, Vic! Good job.
Thanks, Sonia. Okay, bye.
Lots of very good, very positive comments from the chat of people that are living there. Amazing work, really great work. Congrats, great presentation. Okay. So 1 more people.
Yeah, I'll see if I can kick him out. That's my brother-in-law.
That's Justin, right? Justin, go. Yeah.
Yeah. There,
Right.
He's gone now.
Here we go. Okay. Well, the committee is here and the candidate is here. I will say, as an advisor myself, I do not expect to be asking questions. Obviously, if I had questions to Vic, I would have asked them in the process. And so I will leave it to you 3 to ask clarification questions, maybe expand on some of the things that were discussed. I don't know if your dog wants to participate, Vic. And I see that, Tony, you managed to find a spot in a chemistry lab. So yeah.
I'm on the other side of Justin's office, so I'm
you are in Justin's lab?
Yeah, I'm just infringing on everyone right now, so it worked out. Yeah, I apologize again for that. I was in another room and could not get on the internet for like 15 minutes, so thank you, Justin.
I'm sorry about that, Tony. All right, so I mean, you're welcome to ask questions. If you want to ask several questions at once, or if you want to more jump on a question, if it's something that's related to a comment that you had, I think it's the best way, actually. So who wants to start?
I have a question, but I don't need to ask a lot of questions. We could also go to kind of jump back and forth. But I was wondering, And first up, this was amazing, Vic. Such a beautiful presentation, and kind of per usual now, right? We're like coming to like, oh yeah, this is going to be pretty awesome and great slides. And I think you really didn't disappoint. My questions is somewhat related to your thesis, which I thought was excellent too. I really love reading your writing. But when you gave the examples for your weathering reactions, you had the weathering done either just by protons or by a weak acid. And I wondered if you could muse a little bit how that was maybe in the past when strong acids were in the system. Did you kind of think about how sort of the acid deposition times impacted weathering and how rates might have changed, or what do you think might have happened?
Yeah, that's a good question. I think when looking at how the minerals weathered for a year in situ, it was less, we're just looking at that moment, that year, right? So it's kind of, I didn't kind of consider the historical impact of acid rain as opposed to the current impacts of acids in the environment.